Hey people, it's Livestock. Okay? This is from BBC News. So the article goes like this: gene editing to produce super dad livestock. Scientists have produced gene-edited animals they say could serve as super dads or surrogate sires. The pigs, goats, cattle, and mice make sperm carrying the genetic material of donor animals. The researchers used a high-tech gene editing tool to knock out a male fertility gene in animal embryos. The animals were born sterile but began producing sperm after injection of sperm-producing cells from donor animals. The technique would enable surrogate males to sire offspring carrying the genetic material of valuable elite animals such as prize bulls, to the US-UK team. This would be a step towards genetically enhancing livestock. Where was I? Genetically enhancing livestock to improve food production, they added. Professor John Oatley of Washington State University's College of Veterinary Medicine said this can have a major impact on addressing food insecurity around the world. If you can tackle this genetically, that means less water, less feed, and fewer antibiotics that we have to put into the animals. What did the experiment show? The surrogate sires were confirmed to have active donor sperm, and hang on a sec, just got to bring it back up. The surrogate sires were confirmed to have active donor sperm, and the mice fathered healthy offspring that carried the genes of the sperm donor. The larger animals have not yet been bred, but Professor Bruce Whitelaw of the Rusden Institute at the University of Edinburgh said the study provided a powerful proof of concept. This shows that the world that this technology is real, it can be used, he said. We now have to go in and work out how best to use it productively to help feed our growing population. According to the researchers, the technology could also help in the conservation of endangered species. It might be possible to, for example, to use the frozen sperm of an endangered rhino to regenerate the species. But it said the speed at which the science could be put into action will be influenced by policymakers. Gene-edited livestock have yet to be granted approval for human consumption with concerns over product safety, ethics, and animal welfare. What is gene editing? Gene editing involves deleting or changing coding in embryos. An example of a current technology is CRISPR, a biological system for altering DNA discovered in 2012. CRISPR scans the genome looking for the location of a certain gene and then uses molecular scissors to snip through the DNA. While effective in the lab, the process is less than perfect and can cut out too much DNA. These unwanted edits could alter other important genes. What are the ethical issues? The Nuffield Council on Bioethics examining the ethical issues raised by the use of gene editing techniques in farmed animals. Potential applications of gene editing technology include genetically hornless cows and pigs, or chickens that are resistant to diseases. Gene editing could form part of the response to many of the challenges facing societies in different parts of the world, including securing access to healthy and nutritious food, said Director Hugh Whittle. Whether and to what extent genome editing can and should be deployed outside the research setting will depend on further research and development progressing in alignment with societal values and interests that have, in many cases, yet to be clearly defined, he told BBC News. These are among the questions that we are exploring in our current inquiry on genome editing and farmed animals. The research is published in the journal Proceedings of the National Academy of Sciences. I hope you've enjoyed that. Please comment, rate, share, and subscribe. I'll leave this article in the description. If you'd like to follow me on Instagram, I'll leave it there as well. If you'd like to support the channel, leave my GoFundMe there as well. Peace.